now about kind of putting all of that together and speaking about how the membrane potential changes in order to generate this action potential. So let's talk about what an action potential really is. It's a rapid depolarization that is used for cell-to-cell signaling or cell-to-cell communication, specifically excitable cells. So think about neurons and muscle here. And so this is how our neurons speak to each other or communicate signals or messages to each other. They use these rapid depolarization events that can transmit the signal down the length of the neuron, specifically down the length of the axon, and then we can reconvert that electrical energy at the synapse to chemical energy and then start that signal on another adjacent neuron again. So it's just a way for neurons to speak to each other or to communicate to each other, and we can use this electrical energy, this change in the membrane potential, to transmit those signals. And so action potentials travel from the axon hillock, specifically where they start, based upon signals coming in on the soma or the cell body, and they travel all the way down the length of the neuron, and then they are communicated in a different way across the synapse to an adjacent neuron. Okay, now these are the events of a single action potential. So we have the depolarization, which is the rapid positive or upstroke or the increase in the membrane potential here. And then we have the rapid repolarization, which is the downstroke or the negative membrane potential change. Then we have a small overshoot of the membrane potential where it becomes slightly more negative. And then we get back to our resting membrane potential. Okay. Now, as we talked about, sodium is responsible for the depolarizing event. Potassium is responsible for the repolarization. And then what gets us from this hyperpolarization back to rest is actually the sodium potassium pumps that are still on our cell that kind of restore the membrane potential back to -70. Okay. So what we're going to do now is kind of explain this in a different way, and the rest of this packet is just different approaches, different ways to explaining the exact same events, right? The exact same idea of this depolarization, repolarization, hyperpolarization, and then rest, okay? So we're just gonna explain that in a couple of different ways. We can talk about it from the movement of the ion perspective. We can talk about it from the membrane permeability perspective. And as we've already seen, we can talk about it from the gating mechanism on our channels from that perspective as well. So at threshold, we have our -55 millivolts, that requirement being met. That allows the opening of our voltage gated channels, specifically the activation gates on those channels. It also starts the timer for the inactivation gate to close as well. So that allows sodium to rush into the cell, right? That's going to cause that depolarization, that rapid increase in our membrane potential as sodium moves into the cell according to its really strong ECDF. And then the membrane potential becomes more positive as we try to approach the equilibrium potential for sodium. Now, as I mentioned earlier, this is a positive feedback mechanism. So the opening of one sodium channel creates the environment for the opening of neighboring sodium channels. And we'll talk about this going forward, but that is how we get this propagation of the action potential down the length of the neuron, this positive feedback event. If I open up one sodium channel, I'm going to depolarize the membrane potential. That's going to create the membrane potential or the voltage change that now depolarizes the neighboring channel, which depolarizes the neighboring channel, which depolarizes the neighboring channel. And that's how we get the movement of that action potential as we create the voltage requirement that's needed to actually open up these channels. And so we overshoot the membrane potential. Overshoot here meaning that it goes way up past rest, gets to +30, and this is called depolarization, okay? Now at +30, what we see is the closure of the sodium channels, specifically created by the closure of the inactivation gate. So the time that it takes between this -55 threshold being met and the closure of those inactivation gates is that 1 millisecond delay. So I want you to kind of be making that connection of that 1 millisecond delay. So after we have closed that channel, we now open up potassium channels, because remember, potassium channels are also voltage sensitive. So they're going to also open, but at a different voltage threshold. This time it's going to be +30. So when we get to that +30, which was reached or that was achieved because of sodium's movement, we can now reverse those channels. We close sodium channels, open potassium channels, and we can begin to repolarize the membrane potential. And so as we've described earlier, this is a negative feedback event because it takes us from +30, which is really far from our set point, and it helps us to get back down toward our set point. And so we can think about this as being a negative feedback event. Okay. And this schematic here actually very nicely, it has those equilibrium potentials sort of noted to the side here. So remember we said that sodium's equilibrium potential is +60, so way up here. And that influences the direction of sodium's movement, okay, upward. Potassium's equilibrium potential is way down here. And so if I open up potassium channels, that's going to influence the direction of potassium's movement, which is way down here, okay? All right, I'm gonna pause here and see how we're doing with that so far. You can kind of take this in strides here, and then we will see how much more of this we can get through. Okay, so the types of cells that we're speaking about that have this ability to generate action potentials, these are called excitable cells. Examples that I've been speaking about are neurons, for one, that's kind of the main example, but then we have cardiac myocytes or cardiac muscle cells, as well as skeletal muscle cells, which are a slightly different mechanism, but they're also excitable, and they can also generate an action potential. And so this is going to be this large depolarization that we mentioned earlier that can help transmit signals from neurons to heart muscle cells, from neurons to skeletal muscle cells. And so this is really the basis for muscle movement as well. So although we've been speaking about purely neurons, we want to think about them in a more holistic way, the kind of broad functioning that neurons are responsible for. Neurons innervate muscles, they innervate glands, they give glands the instructions to secrete or produce their secretions, they innervate muscles, they give muscles the instruction to move. And so action potentials are vital, very, very important to the nervous system function, to muscular system function, to even the functioning of our glandular tissue as well, okay? All right, and so let's look at a few checkpoint questions here in the final minutes and sort of recall some of this. So the first question is, define depolarizations and hyperpolarization. So how would you define or describe a depolarization versus a hyperpolarization? Any ideas, any thoughts here?
Depolarization is when, I'm trying to think of the best way to phrase it.
It's okay, take your time.
It's when the active channel and it's when the active gate and the inactive gate are both open, and in the context of sodium, it's allowed to pass through freely. And it's when the charge goes from -55 millivolts to 30 millivolts.
Excellent, excellent, well said. So you explained what the actual value was, right? So it's a positive membrane potential change. We go from -55 upwards to positive 30, but then you also explain the why, right? Which is the idea that we have both of our gates open on that sodium channel. Sodium can wash in, and that is what creates that membrane potential change, okay? Excellent. And then hyperpolarization is going to be the opposite, right? And it's different from repolarization. So although repolarization is not listed here, hyperpolarization is going to be a negative membrane potential change, but one that moves away from set point, okay? It's going to be a negative change in the membrane potential that moves us away from that negative 70. So it's going to be more negative than negative 70, if that makes sense. Number 2 here, if the permeability of a membrane to sodium increases, would the membrane depolarize or hyperpolarize? And I think that kind of ties into what we just described, right? If the membrane permeability increases for sodium, that is directly responsible for the depolarization, which is a depolarizing event, okay? Same thing. Number 3, name the 3 gating mechanisms for ion channels. So what are some of the gating mechanisms we described earlier? Basically, what are the 3 types of ion channels? So in the interest of time, I'll go ahead and kind of explain that here. So we have no gating mechanism. In other words, it's a free flowing, open, or a leak channel, right? We've got the voltage gated channels, and within that, they can be voltage dependent only or voltage and time dependent. So 2 requirements for that voltage, for that opening of that channel, for that mechanism. And then the third type were the ligand-gated channels. And those are the channels that are found exclusively on the soma and the cell body along with the dendrites. And those are the ones that have to have something bind onto it, some type of ligand. The neurotransmitter is the example we spoke about that will then cause that channel to open up. So we have 3 different mechanisms. Either there's nothing that's required, either a ligand is required, or either a specific voltage and time in some instances is required. And then the last question, what is the threshold value for an action potential? We've said this quite extensively. It's negative 55 millivolts, okay? When we get to that negative 55 millivolts, we must fire a complete action potential. There is no gradation. There's no such thing as half of an action potential, 3/4 of an action potential. It's either a full action potential or no action potential, okay? And we'll talk about that when we meet on Friday.